for Genomics, ICG, both at Peking University. Sunney received his bachelor's degree in chemistry from Peking University and PhD from physical chemistry from UCSD. And after a brief postdoctoral appointment at University of Chicago, he joined Pacific Northwest National Laboratory, where he rose from senior scientist to chief scientist. And from 1998 on, he became a full tenured professor at Harvard University, where he stayed for 20 years. And he's a foreign member of the Chinese Academy of Sciences and a member of the U.S. National Academy of Sciences. Sunny, are you ready?
Yes.
Okay, great.
Oh, thank you, Zeming. Hi, everybody. The COVID-19 pandemic has proven catastrophic for public health and the world economy. This morning's death toll of the world is over 1 million. So I'd like to share with you what we have learned and accomplished during the past few months. It has been an emotional and stressful time as China and the world we're dealing with the spreading virus. But as scientists, we all try very hard to make a difference. You all know that many vaccines are under phase 3 clinical trials. Before safe and effective vaccines are widely available, there are always going to be COVID patients who badly need effective drugs against the disease. So here I have a recent review of all the anti-COVID-19 drugs. It's probably very hard to see, but just focus on this last column. The bottom line is few are effective. We certainly don't have an effective drug at this moment. So what would be an effective drug to cure COVID-19? It would significantly cut down the proportion of patients with mild to moderate symptoms turning into severe symptom. It would significantly reduce the damage of patients' lungs and other tissues. It would quickly cure mild cases, thus shortening the hospital stay. Now, you all know that the convalescent plasma therapy has proven to be effective in clinical trials, but it is limited in supply. One patient needs 1,200 ml of blood from recovered patients. So the supply is limited. Neutralizing antibodies are the active components, ingredients of the convalescent plasma. And they are produced by the human immune system, B cells. So our goal is to identify the neutralizing antibodies from convalescent patients and use them as a substitute to inject into COVID patients. So this is a SARS-CoV-2. It is RNA virus. This S protein on the surface is a trimer. It has this receptor binding domain, RBD, in red color here. Chinese scientists determined the genome sequence of SARS-CoV-2 in a matter of a week or two and reported to the world. And that provided the basis for diagnosis and therapy. And then a few groups, including 3 Chinese groups, reported the structure of the S protein with RBD, and later on its binding to the ACE2 receptor that is on the surface of the cell membrane of human cell, such as ones in the lung. So neutralizing antibodies binds to this S protein, and that would prevent the binding of the virus to the ACE2 to the human cell to infect the human cells. Now, these are the previous success stories of neutralizing antibody, in particular HIV. In 2018, the US FDA approved the drug for the treatment of multi-drug resistant HIV in combination with other antiviral drugs. In the case of Ebola and MERS, antibodies, successful antibodies, were also developed, but the pandemic, the outbreak was ended before the clinical trial were finished. I particularly want to mention the work of Michel Rosenzweig. He first looked at different individuals of HIV patients using single-cell clonal amplification. You see these 4 different patients. They have a different number of antibodies. The ones in green color are not effective, not potent, and the one in other colors are potent. But you see, different people have different. This number 4 has many colored ones compared to this one. So this really highlights the importance of why we want to do single cell. Suppose you have a patient, its blood has a high potency in neutralizing the HIV virus. How would you know it is because it has a large number of mediocre antibodies, or it has a small number of highly effective antibodies? So we must determine the distribution. I think this is really anybody in our community understands this. But in order to do this, the old technology was a clonal amplification, which is highly time-consuming. Now, in recent years, many groups have been pushing for this high-throughput B cell sequencing, in particular Steve Quick's group, and others. These are just 4, I think, a really good paper in the past year or 2. So basically, it's a high throughput sequencing and give us the speed. Now, my own group were not doing this at all before the pandemic. We were really forced to do this when Wuhan was closed down, and we had to. The technology was ready, and we had to put it into human use for real. Doing it in a very urgent way, and we were racing with time, and it worked, and worked very well. And of course, that's after very hard work. Our effort started the 3rd day after Chinese Lunar Year. Richard Cao, who was the only person who moved with me from Harvard University to Peking University, he arrived last year. We arrived home on the same day after the break. And at the beginning, we were working on diagnosis experiments. But when we realized that the single cell approach would really accelerate the process, allowing us to find effective neutralizing antibodies, we were very excited. We started reading literatures, borrowing equipments and reagents, and everybody were extremely helpful. And then only a few days later, we went to Yuan Hospital, one of the designated hospitals in Beijing for COVID-19. We met President Jing there, who immediately agreed to provide us the convalescent blood. And then he sent 2 researchers from the hospital, Su Bing and Guo Xianghua, to our sequencing center. This is our sequencing center's manager, and Richard and Cheng Yang trained them too. And then they went back to their hospital's P3 laboratory, wearing this heavy protective gears. They were actually only allowed to work 5 hours normally, but they always work for a long time, and they were isolated for 2.5 months. On our side, Richard and Chen Yang were fighting day and night, sequencing the convalescent blood from recovered patients. So this is the RNA sequencing, and of course we see the different cell types. This is the development of the B cell from bone marrow, and as it gets matured in going to the blood and lymphocytes. So we were most interested in the memory B cell and the plasma cell. Now, at the beginning, each B cell undergoes this VDJ recombination. I think most of you probably know this work won the Nobel Prize in 1980s. So this genome here on chromosome 14, this V sequence got 40 of them, and D 23 and J 6. And then this VDJ recombination discovered by Tony Gava. So you pick one V sequence and one D sequence and one J sequence. It's like you select a deck of cards from this 40 and then from this 23 and one from the 6. And it just this combination would give you the diversity 40 multiplied by 23 by 6. That's 5,000. That's still not enough. The diversity is huge. A human body would have 10 to the 7th, 10 to the 8th different types of sequences combination. Now, that's because in addition to this number 14 chromosome, there's also number 2. That's for the light chain and heavy chain and light chain. And that's still not enough. This point mutation, in addition to the VDJ recombination, give more sequences. And these different sequences result in different proteins with different molecular interactions, different binding affinities to various antigens. So what's interesting is this 10 to the 7th or 10 to the 8th different type of proteins, they exist before the any antigen enter the human body. So these cells do not divide, but on top of the on the surface of the membrane, this B cell receptor, BCR, it's like a Y-shaped antibody, except it's linked to the membrane. They're there until, so when antigen comes in, such as our SARS-CoV-2, it binds to this BCR, and then it activates the B cells such that they start to proliferate, the divide, and one becomes 2 to 4. And then Some of them become the plasma cell that secretes the antibody, and that becomes actually the neutralizing antibody that neutralize the virus. And some other cells become memory B cells. On the surface of the memory B cells, there are this PCR, a particular type of PCR called IgG, and that's what we're interested in. We want to isolate memory B cells with the PCR on the surface. Now, actually at the beginning, what we did was we just used the 10x with no functional selection. So we, because this high throughput, we sequence many cells, and notice this 6 cells, they have identical VDJ. In the VDJ region, they have almost identical sequences. That means they all came from the same parent cell. So they have the same, they are enriched, they are amplified, they are after the activation process I just mentioned. So our presumption is, of course, the higher the enrichment, the more useful this particular sequence to the immune system is selected by the complicated evolution process. Right. So once we determine the sequence, we of course can use this sequence to synthesize the protein in vitro according to the central dogma. We actually ask a company to do the in vitro translation. In doing that, we get recombinant proteins, and we do the testing for the functional assay to detect the binding affinity, the neutralization potency. We believe it or not, even on the first patient, we were able to find 1% of the this enriched sequences resulting in antibodies that have neutralizing ability. But of course, 1% is very low. We want to improve that. And so we combine that with the functional selection. We use the S protein linked to the magnetic beads, and this magnet basically you separate this memory B cells with the BCR. And later on, we also use RBD only on the magnetic beads. So this gives us the high throughput results, which we published in July. We had this within 2 or 3 months. We had 8,558 IgG sequences that are that were as the candidates. And I want to stress that compared to any other groups, I think right now many people use this approach, but at the beginning, this number is order of magnitude higher compared to any other groups, and this high throughput really give us the ability to select better neutralizing antibody. It's simply a probability problem because there are 10 to the 7th, 10 to the 8th, and no matter what we do, we sequence 10 to the 5th using a 10x box, for example. Then we're only sequencing a very small fraction of them. So the more we sequence, the better chance we find the good ones. So at that time, we found 14. Now we have 20 highly potent neutralizing antibodies. And what we found is a really good one. And let me show you this IC50 curve, the neutralizing curve. The horizontal axis is the antibody concentration, and the vertical axis is the percentage of the cells that are neutralized. So this middle point, IC50, but you can have IC80, IC90, and so on, if you want to see how steep this curve is. Of course, this IC50, the lower, the better. This corresponds to the amount of drug that you have used. Some of you probably know that Trump received 8 grams of antibody, and I think this number is high compared to what people normally do. In the case of cancer, PD-1, PD-L1 is usually under a gram. So if there are highly potent neutralizing antibodies, one could have lower dosage. Xiaodong Su and Junyu Xiao, 2 structural biologists at Peking University, they determined the structure. This is a cryo-EM structure. The bottom in light blue is the trimer, and the red is the RBD, and the pink being the antibody. Now, this antibody binding really blocks the binding of the S protein to ACE2. And we found most of the potent antibodies binds to RBD from the crystallography, but even from the comparison between the RBD pulling down and the S protein pulling down. Okay, neutralizing antibodies have actually 2 uses. You could use it as a therapeutic drug, or you could use it as a prophylactic temporary prevention for patients, family members, or medical workers, or somebody wants to go out for a trip. But the effective time for the protection would be the lifetime of the antibody, which is we measured to hours are 28 days, which can be extended if we modify the protein. So Dr. Qin Chuan's laboratory established the humanized ACE2 transgenic mice. So the animal experiments were done in her lab. So we at time 0 let the mice get infected by SARS-CoV-2, and then 2 hours later we give this 386, our most potent neutralizing antibody. So that's the therapeutic experiment in green color. And then this the blue is negative control. So 2 hours later, we give it a irrelevant antibody. So after 5 days, the viral amount in the lung is very high in this negative control, and this blue one is reduced by a factor of 2,500. But if we give the 386 2 days prior to the infection, then we actually detect no virus in the lung. This is the body weight as a function of time. You see in the case of the therapeutic and the prophylactic, there's no weight change. There's a slight going down, but even if you just give a shot, you would reduce the weight. So this was reported in our first Cell paper. I want to tell you the result of our second Cell paper. Junyi and Xiaodong have more interesting structural data, and Qinchuan's laboratory has done this on hamster, which really give us a feel on the dosage, and then we develop a new cocktail. So you see, this is the 386-2, which is potent than many of the other antibodies developed by our group, other groups. So in this Quirl-EM structure, this is the trimer of S protein. There are 3 RBDs, 1 up, 2 down. And their structure shows that our 386-2 bind to all 3 of them, no matter it's up or down. Whereas the first structure we got was only bound to 1 of them. So this explains the high potency of this neutralizing antibody. It really it has a different stoichiometry, and this was further evidenced by the ELISA binding. So this provides a structural basis for the extremely good potency. Okay, then Qinchuan's lab used hamster model, which is advantageous over the mouse model in that the hamster is heavier, and it can actually get infected more severely or even die of COVID-19. And so we noticed the first time we had this dosage of 20 milligram antibody per gram. So now we have under this dosage, we first look at the window of dosage. At the beginning, what we did before was 2 hours after, right? So now we extend it to 48 hours. You see, hamster and mice, their metabolism are much faster than humans, so everything is accelerated. So this multiplied by 7 roughly is the human time. So that's about 2 weeks. So this corresponds to moderate. It's not a heavy symptom. The heavy symptom actually is another disease. The immune system is totally wrong, right? So, but extending roughly to 2 weeks, this is the moderate symptom. Now, this was by weight. Of course, the most rigorous way is by the pathological results, which is consistent. So that's the dosage window. We also changed the dosage. We went from 20 milligram per. It's a 20 milligram per kilogram. I dropped this kilo. Sorry. And then we went to 10, 5, 2. Now, of course, the lower the better, as I mentioned. And then again, the pathological results is consistent with the body weight result. Okay, so this is a really good news. It is consistent with the fact it is highly potent. So finally, I want to briefly mention the pairing of antibodies of non-overlapping epitopes. This is the structure of 2 antibodies. They don't have overlapping epitopes, and then when we use them as a pair, then some of the strains that has undergone mutation, well, luckily these mutations are not very abundant, but when we apply the pair, they could avoid the mutation escape. And this is our latest result, which is very interesting. You see, if we just use one of them, one alone, you will have IC80. This is similar to IC50 at the beginning. Okay, so these are pretty low, but after even 2 days, the IC50 increase. That means the drugs are not very effective. Why? Because under this antibodies attack, the evolution pressure allow the system undergo mutation, and then the drug become non-effective. But if we use the pair, you see the IC80 does not even change on the 5th day. So to end, I want to mention that these 2 students of mine formed Single Omics, a startup developing this antibody drug. I mentioned this as the disclosure of my potential conflict of interest, just to for the sake of complete disclosure. And Single Omics and Beijing, a drug company, are collaborating, developing neutralizing antibodies, doing the clinical trials around the world. And here I have a list of the clinical trials of neutralizing antibodies around the world we know. So this is Regeneron's and Eli Lilly. They're ahead of the beginning of the game. And so let me just quickly summarize what I have told you. High throughput single cell transcriptome and VDJ sequencing speeds up the development of clinical use neutralizing antibodies. So we have found 20 potent neutralizing antibodies from this 8,600 B cell VDJ sequences from about the 60 plus convalescent patients. Where EM structures show effective neutralizing antibodies binds to the RBD of spike protein. And the most potent ones binds to all 3 RBDs, no matter it's up or down, in up or down conformation. And a pair of neutralizing antibodies with non-overlapping epitopes can effectively avoid mutation escape. Our animal tests demonstrated that these neutralizing antibodies exhibit high therapeutic and prophylactic efficacy against SARS-CoV-2. I wouldn't be able to tell you the stories if I did not work with a group of very talented, hardworking individuals. Many of them I got to know only after the pandemic. The institutions involved, and I want to thank the support, mostly from Beijing municipal government, and I want to thank you for your attention.
Thank you very much, Sunny. That's really amazing progress, given that the project didn't even exist at the beginning of the year. So that's, I have time for a few questions, but let me take the opportunity to ask this question. So you generated 8,558 antigen binding clonotype. That part, I imagine, is relatively easy because you go through this whole large-scale single-cell sequencing. But then from that point on, you identified 14 neutralizing antibodies. So how many did you have to test before you find this 14? Later on, you mentioned 20 neutralizing antibodies. I imagine that requires a lot of individual testing.
Well, we've synthesized a total of 600, almost 700 peptides. But that's because we didn't know what we were doing. I mean, if we were to do it again, I don't think we need to synthesize that much. I mentioned at the beginning, the yield was 1%, right? That is, if we synthesize 100 of them, one would would have the potency, and that's because we didn't have the functional selection. As soon as we have the RBD pulling down, S protein pulling down gave us like 20%, and then RBD gave us 30%. That is to say, if we test 10 proteins, the first is ELISA, the binding, but just the binding doesn't mean it's functionally active. So we do this pseudovirus assay in our lab, and we once confirmed it is potent, we also do the authentic virus in the P3 lab. And then later on, we use the structural information, those protein sequences that already have protein data bank structure for, say, previous SARS. We found then many of them are like that, and that would give us 80% or even 90% of the yield. So, so of course, if we know nothing about the antigen, yes, that's like a blind search. But that's what we had. We didn't know. That's what we had at the beginning. But you must, if you know the antigen, of course, it will be just much faster, and pulling down with antigen give us this high yield at the end.
Okay, let's. Next question comes from Gary. Gary Nolan, do you want to?
Hey, Gary.
Hi there. Yeah, so I'm not the first person to suggest the idea, but have you tried putting any of the sequences into lentiviruses or even more short-term viruses to be able to infect them into target cells so that they would produce for a longer period of time?
To put it in the
say infected into macrophages, so cells that might last for 2 or 3 weeks, or even frankly, semi-purified B cells from the patient. Put a single chain version of it in.
For what purpose?
To produce the antibody for a longer period of time rather than having to inject it. Or, for instance, putting it into the muscle of a patient.
So actually, an easy way to prolong the lifetime, if that's the goal, the modification of the FC region would easily get to 3 months or so, and some people even talking about half a year. Yeah, so we didn't quite do that, not yet, but actually 3 months is probably
3 months is pretty good. I mean, but if you don't have to inject every 3 months, that would be better.
And the difference between this and the vaccines are the vaccines would, of course, have a memory, and there's this possibility of ADE, antibody-dependent enhancement. So when the let's say if the virus undergoes mutation, say next year it gets really bad, so much such that the antibody would not be that effective, and simply having these antibodies but not doing their job anymore, they would bind to the virus, and then they would help the incorporation into cells, because the FC, the cells have this FC receptor. So that's the ADE. In fact, one of the theory of why the Spanish flu, in the second year, a lot more people died after the almost the entire population. The first year is like now, 1 million people died. So that was a theory. Of course, nobody knew what was really going on 100 years ago. So that's a possibility. And the neutralizing antibody doesn't involve the memory, but it is reasonably inexpensive, the prophylactic use. Actually, AZ is doing the clinical trial. The clinical trial is of course harder than the therapeutic, and AZ is doing that for their pair. And actually mentioned Regeneron was really the first company that pushes the pair, and Trump used 8 grams of Regeneron's pair.
Okay,
great. Thank you.
So we have a next question that comes from Shu Zhang.
Hi, Sunny. I have a question about the antibody structure. You said that in the first Cell paper, you said that you find 8,000 clonal types. I want to know if they share some efficient structures that may help scientists synthesize the antibodies.
In the first Cell paper, we really just reported one antibody's structure. But in the second paper, we have a lot more structures, and we actually had a very important finding. Actually, this finding was reported in the first paper. We found so-called convergent, or some people call recurrent antibodies. You see, this diversity is huge, right? So yet we found different people, different individuals have identical structure, identical fold. The reason of this, it's not absolutely clear, but the finding itself. In the second paper, we reported the structures. Basically, it's structures from different labs on top of each other. There are a few of them. You see, I just want to show you this 629 is this one, right? All the other convergent antibodies, they all bind here from different labs. So this 368-2, it is special, it's on this side. So the good thing is it doesn't overlap, so they can be used as a pair, but all the other ones, 10 times lower, they're on this side. So the 604 that we're going to use with Beijing is actually very similar to 629. So they are convergent. They have identical structures almost.
Okay, thank you.
Thank you very much, Sunny, for the amazing work and talk. Next, I'm going to introduce our very